Yeah, let me have a look at the data. Okay, so last week you are supposed to cover medium formulation and optimization. We are still talking about fermentation in general. So yeah, I will record the manual attendance later. Okay, we are still talking about fermentation process in general, because this course is concerned with upstream and downstream, remember? And now we are focusing on upstream first, and fermentation is part of the upstream processes. Okay, so we talk about fermentation in the first week. Yeah, what is fermentation? It's a production process in biotech, and why it is important? Because why this field, bioprocess, is important? Because this field is the one that connects whatever the discovery in the lab and also the factory or industrial scale. So without bioprocess technology, we're not going to have any products reach to us, yeah? Like the vaccines, the antibiotics, enzymes, the fermented food, and all this stuff. They have to go through all the bioprocess field before they reach the consumers. So that's why we have to study bioprocess technology. And part of the bioprocess technology is fermentation, because fermentation is a production process in biotech. Okay, and we have talked about it in the first week. Yeah, what is fermentation? What are the main components of fermentation? And what makes a fermentation process? Yeah, there are various types of it, various formats, and various platforms as well. So in the second LU, in the second week, you are expected to cover medium formulation and optimization. What is a medium? For microorganisms during the fermentation, they are food, right? But in order for us to make the microorganisms grow happily, we cannot just provide food just like that. We need to formulate. We need to create a recipe, and not just formulating, but we also have to optimize. Formulation and optimization are two different processes, but they are related. First, of course, we have to formulate. We have to create a recipe. What are the components that we want to add in our recipe to the in our food to the microbes, right? And that process is formulation. Yeah, formulation. And then after we have formulate, we know that the microbes can eat this, can eat that, and they are not allergic to certain components. Then we have to proceed with optimization. Optimization is a process whereby we continue from the formulation. We try to find what's the best concentration of glucose, what's the best concentration of nitrogen source, the trace elements that can make the microbes grow much happier than it is if, let's say, we just give them, like the basic level of food. Right? So that is optimization. So I hope that you have read the story, the Ahmad story, right? When if you notice that when he tries to optimize, he vary the concentration of the glucose. Because in the first place, he knows that his microbes can eat glucose. Not every microbe can eat glucose. Glucose is a type of carbon source. Carbon source is, like it's carbohydrate, like as we eat rice, right? Rice is a starch. So starch is made from glucose as the monomers. So for the microbes, some of them can eat glucose, some of them are allergic to it. When I say allergic to it, it's like humans. When we are allergic to something, what happens? We may form some sort of irritations on the skin, things like that, and at a certain point, it could cause death. Right? So same goes to the microorganism. If they are allergic to something, it means that they are not happy with that food, and it can eventually lead to death. Yeah. So that's why we need to identify what are the things that they are allergic to, what are the things that they are happy with. So optimization is something to do with like finding the right concentration, the right amount. Let's say, for the medium components, we quantify it in the form of concentration, gram per liter, how much gram per unit volume. So we vary the concentration from the lowest to the highest, and at certain point, there is a point where that is actually the limit, the maximum limit for the microbes to bear with for that particular food. So we need to identify that point. So that's why we vary it from the lowest to the highest. Yeah, so from there we can know at which point that the microbes will be it can sustain with. So how to know that? During the fermentation, we have to monitor the growth. We have to take the samples, check the growth, and also check the amount of products produced. So of course, before that, we need to know what are the products that the microbes produce. So that is the first motivation why we grow them. So from that quantification, and only then we know that they are happy or not with the food that we supply. Okay, so that are the things that you are expected to cover in the second LU, medium formulation and optimization. And I have asked you some questions, in the activity. Question, 9 questions. Let's go through briefly. First question is about what is media? What are media to the microorganism? Yeah, they are food. They contains like all the important nutrients. Nutrients foods are the same, okay? But in the lab we call media. So why we provide it? We want our microbes to grow, and because we bring the microbes to the lab. Lab is an artificial environment. It's not their natural environment. They are naturally thriving or growing in their environment, like soil, in the water. So when we take them to the lab, we have to provide artificial food, and those that we provide in the lab are actually artificial food, like it's not their natural food. Okay, right. So that's correct. Okay. Group 3 media. Where's the answer for group 1? Group 3. Media of food. Okay. Second question, what is the purpose of isolation of microbes from their natural environment? Okay, remember in the Ahmad story, Ahmad collected the microbes from the water, right? So my question is related to that. What is the purpose of isolation of the microbes? Why Ahmad took the microbes from their natural environment? So to answer that, the answer is actually because Ahmad has done some literature review, and he knows that maybe in water there are some bacteria that can produce certain beneficial products, right? So that is based on the information that he got from his literature review, for example. So in general, the purpose of isolation of microbes from the natural environment, because we know that certain microbes, they can produce certain useful products. So that's why we bring them to the lab. So those microbes that maybe do not produce the one that we want, if we want, so we might not bring it to the lab. So the purpose is because those microbes have certain functions. They have certain roles in producing certain metabolites that are needed by humans for solving problems. Remember, biotechnologists, we need to solve problems a lot, right? So we need the microorganism, we need the biological organism to solve the problems for us. But of course, we have to, to grow them effectively in the lab. Okay, so that is the purpose of isolation. Isolation means like taking the things from their environment. Okay. Question number 3. Okay. So once you collected the microbes from their natural environment, we cannot see from our naked eye that this is E. coli, this is lactobacillus, this is bacillus. We cannot see from our eyes because they are so small. They are microbes. They are their size is at micro level. That's why they call microorganisms. So what we have to do is we have to carry out certain identification tests. So that is the question, right? The microbiological tests. Remember in your first year when you take microbiology, you study about different tests that you can carry out in order to identify the identity of the microbes that you are examining or you're isolating. Okay, so basically the objective of those tests is to identify the microbes. and it's based on certain redshift, right? I mean, you have to do, sometimes you have to do a series of tests whereby you can combine, you can complete the puzzle. Like this one, it gives like maybe pink, gram staining. So maybe it's a gram negative. And then when you examine under the microscope, you see that the shape is maybe in rod shape or in sphere shape. So you combine the puzzles, and at the end, you know that, okay, this one match this one match this, so it satisfies the identity of maybe E. coli. So that is actually the purpose of it. So at the end, you want to identify the identity of the microbes, because you cannot actually identify without doing those things. So that is actually the objective of the microbiological test, that is to identify the microbes. But of course, yeah, in many applications, you will relate it with, for example, if let's say your objective is to identify pathogens, you want for the food safety. That one is in the specific application. But the general purpose of it is to identify, to detect, yes, to know what are the microbes before you can grow them, because you need to know them first, right? And then once you know it is E. coli, then you have some ideas about E. coli based on the reports, previous reports. Okay, E. coli can thrive at 37 degrees, and then it can eat glucose. So that is the importance of identification. Without identification step, you cannot know what your microbe is and how you're going to provide the food. Yeah? So that's why I asked the question. Okay, question 4, what is the medium formulation that is important? Okay, I've talked about it. It's about creating a recipe for the microbes in the lab, because we need to create a recipe that can make them happy in the lab. So it's about creating food for them, formulation. Its importance is because we want to grow our microbes in the lab. Okay. Okay. Question 5, what is synthetic medium? And what is the difference between synthetic medium and natural medium? So synthetic medium are those that are created, that are not natural. Natural medium are those, for example, like the microbes, when they grow in the natural environment, whatever they consume, that are natural, because that are, they are in the environment. So that are natural, for example. And for synthetic medium are normally those that we use in the lab. Glucose. The glucose is produced, I mean, there is a process of producing it. So that's why it's called synthetic. And then it's not just glucose. There are so many chemicals that we use, right? The trace elements, the nitrogen sources. So all of them are produced at the factory, and then we buy it and bring it to the lab, and we use it for the media, because we know that glucose can be used for growing the microbes. So those are synthetic medium. So and of course we know what other, like glucose, we know the components, because they are manufactured in a known procedure. But for the natural media, if let's say you talk about soil, that are natural, but in soil there are so many things. We do not know what's the exact components in the soil. Of course, if let's say you examine it, you know it, but in general, you do not know how much is this, how much is that. Okay, so that is one of the big difference between natural medium and synthetic medium. But in the lab also, we can use natural medium. We take some things like from the natural environment. Yeah. For example, like agricultural waste, there are examples of natural media. But in order for us to use those natural media, of course, we have to do quantification, characterization. We need to know how much of whatever the components are there, in order for us to optimize our fermentation when we use them for growing our microbes. Okay, basically, natural media and synthetic media, they differ in terms of the components, in terms of the origin. Okay. But in the lab, we can use both. We may begin with synthetic media first, but then after that, let's say we want to cut the cost of the production, and we found out, let's say, some agricultural waste have some benefits to be used as media. So we can use like maybe sago hampas, or maybe the waste from the fruits that are rich in glucose. So that are examples of natural media, but both are applicable actually. Okay. Question 6. What's the question 6? Okay, some of you answered. Yeah, you got some of the points. Okay, for the 5 questions just now. Question say, what is the purpose of pH adjustment and autoclave during medium preparation? Yeah, optimal pH is required for microbial growth. Yes, correct. And then autoclaving is for? Yeah, it will actually kill whatever the microbes that we don't need, because we need the media to be sterile, to be clean, so that when we have our microbes, it's only our microbes that can grow in that media. Okay, that's the purpose of autoclaving. Another group which will answer correctly for that. Group 7. Group 6, is it? Okay, where's the answer for group 6 here? Okay. Purpose, neutralize chemical, provide optimal. Yes, okay, correct. And then pH adjustment, that is just some autoclaving. pH adjustment, okay, when we prepare media, that media actually has certain pH values. So of course we have to adjust the pH, because if let's say we know already what's our microbe is, let's say Saccharomyces cerevisiae, and we know that Saccharomyces cerevisiae can grow at pH 6 to 7, for example. So we have to make sure that the pH of our media lies between that values as well. If let's say our pH, our initial pH of the media is, let's say 3 to 4, okay, so we have to adjust it to 6 to 7 in order for it to be able to be eaten by the microbes. Okay, so that is the purpose of pH adjustment, because we need to cater to the fabric environment of the microbes. Question number 7, effectiveness. Oh, yeah, effectiveness, I've mentioned about it. So how you want to know whether your microbes are happy or not with the media? You have to examine their growth. You have to take samples, check their growth using certain methods, normally using absorbents for bacteria and yeast. And from there, you can know that whether your microbes are growing or not. And then for the products, it depends on the products. If your products is if the products of the microbes is bioethanol, then you have to check using suitable methods about the level of the products produced. It means that when the products are there, the microbes are growing. Okay, so that is, it means that they are happy with the food provided. Okay, that's how you know, that's how you assess the effectiveness of the media. Question number 8, glucose. What's the question? Glucose is a carbon source. Okay, carbon source. And then peptide is a nitrogen source. Okay, that's the answer. Glucose is a carbon source, and nitrogen is peptide is a nitrogen source. Group 8. Yes, you correct. You answered correctly. Okay, question number 9, media optimization. Okay, so I mentioned after formulation, the next stage is optimization. Optimization is you want to know what's the right level, what's the right concentration or amount of the medium components. Because when you formulate it, you have like glucose, you have peptide, you have trace element, but you might need to know also how much glucose that you need to add. Is it 10 grams per liter is enough, or how? And also goes to the rest, nitrogen source like peptide, how much peptide that you need to supply. So that has to be done through optimization. Okay, that's the importance of it. Okay, so enough for LU2. I hope that you have understood the concept clearly. Okay, let's just go directly to LU, today's LU that we're going to cover. So we are still talking about fermentation. And today we are going to discuss about design, features, and process control of bioreactors. Okay, so I think you have come across the word bioreactor. Bioreactor is a place where we carry out the fermentation. Okay, so these are the subtopics that we are going to cover. First, we're going to have a look at some introduction, and then types of bioreactor. There are few types of bioreactors, and then fundamental of bioreactors, sterilization, and also forming. Okay. Now let's recap. What is fermentation? Okay. Fermentation is the process of growing the microbes. Why we want to grow the microbes? Because we have an idea that the microbes can produce our product of interest. So that's why we grow them in the lab, right? And we grow them in a place, in a suitable platform. Okay. So the first important component of a fermentation is the microbes. And then what we supply to the microbes, the medium or the media, or food. So we have to provide suitable food for the microbes, and only then the fermentation can take place effectively. Okay, so you know about formulation already, you know about optimization. And then the next part is what we're going to cover today, which is the platform and conditions. So in order to carry out the fermentation, we have to put the microbes, put the media in a closed system, in a closed place, whereby we call it platform, in a suitable platform, right? And that platform, in general, we call whatever the place or the platform that we use to carry out fermentation as fermenter or bioreactor. Fermenter, bioreactor are the same. Okay, sometimes I would say fermenter, sometimes I would say bioreactor. So please take note that both terms are interchangeable. So they mean the same. So that's the thing that we're going to cover today. And not to forget, our objective of fermentation is to produce products, because we know that certain microbes can produce certain products. And that is actually the ultimate aim of a fermentation process, to get the products. And that's why the fermentation process is a production stage, actually, is a production process. Yeah? And it is part of the upstream process. So remember, bioprocess is divided into upstream and downstream. So upstream is concerned with the production. So once we produce the products through the fermentation process, we need to pass the products to the downstream. If let's say in the factory, they have to pass it to the downstream unit, DSP, downstream processing unit, for the purification and for the separation of those byproducts in order to get the main product only. Okay, but that's going to be discussed in, I think, LU 6, yeah, in later LU. Okay, but now we are still in the upstream stage, in the fermentation stage. Okay, let's recall from the materials that I gave you last 2 weeks. Did you remember what platform that Ahmad used for his fermentations in the lab? Could you please scan the QR code, or you can go to menti.com and use the code given here and answer the question. What platform did Ahmad use for his fermentation? It has to be a very short answer. Don't answer too long. Any answer? Oh, you have to join this. You have to go to this menti.com. Biofuel? What platform did Ahmad use for his fermentations? Platform. Okay. I'm waiting for your answer. Please answer now. Okay, I need to answer. How many of you in this class?
Doctor,
Yes?
can I have the code again? I can't see the code just now.
The code. That one. Can you see the screen now?
Yes.
30821092.
All right, thank you, doctor.
Yeah. You go to menti.com, www.menti.com. The code is here. You can see from here. 30821092. What platform did Ahmad use for his fermentations in the lab? Come on. You can answer even though that the answer is there, or maybe your friends have answered. You just answer based on your understanding, because I need to know how much of you understood the question, how many of you understand from the story, the Ahmad story. I'll give you 5 minutes more. Three more minutes. Is that all? Look at that. This one is just that's PowerPoint. Okay, any more? 20 seconds. What platform did Ahmad use for his fermentations in the lab? So if when you read the questions, you have to understand it first lah. What I'm talking about here, fermentation. Fermentation means it needs platform. It needs a place for you to grow the microbes, right? Platform in plain words, is any place, right? So from there you can imagine that it is a confined place. Confined place. So the answer here is shake flask, right? So why I see so many answers like media, journal papers, petri dish or petri dish. No, it's shake flask. Ellen Mayer flask, yes, correct. Shaker flask, shake flask, boleh lah. Shake flask, Ellen Mayer flask. Okay. So why some of the answers are not related? Continuous platform, microbial platform, media, culture media. Media is other thing. Is platform is a place. Okay. Fat bash is a technique. I didn't mention about fat bash. Continuous synthetic media batch fermentation. Okay, so the answer is LMA plus or shake plus or shaking plus. You can also say that. Okay, it's a platform. Okay, remember? Okay, I hope that I'm quite scared to see the answer. It looks like you don't really catch what I really talk about in my first lecture, in the second lecture. Okay, let's go back to our lecture. So the answer is shake plus, yeah, shake plus. If you go back to the materials, you can see that I might grow the things in the shake flask in the incubator shaker. So the incubator shaker is not the platform. So the one that where the microbes are actually, that's what I mean by the platform. So the platform, yeah, this one is where the things are, okay? So it could be various actually bioreactors, but for Ahmad's story, he used shake flask. Okay, that's the answer for it. So bioreactor by definition is a place where the bacteria or the microbes in general are grown for their products. It is an equipment that provides a control environment in order to achieve the optimum growth. So it is a closed system, right, in the lab, because we do not want the microbes to be exposed to the environment. So that's why we have to create that platform. So bioreactor, by definition, is where the fermentation takes place. Fermenter or bioreactor is the same thing. So by definition, if we say the shake flask is also a bioreactor, but in the lab, we don't call shake flask as a bioreactor, although by definition it meets all the points here. But because of in the lab, we have another equipment, advanced equipment that scientists have invented, which is called which is actually a vessel consisting of the control units, and that is what we call bioreactor in the lab. Okay, so yeah, so by definition, although bioreactor, it can be any place, any platform, even your guts, your perut, stomach, can be also a bioreactor because it carries out the fermentation. Okay, but in the lab, when we say bioreactor or fermenter, it refers to the specific machine or vessel. Okay. Right. So shake flask shake flask is also, they are also used as a platform for fermentation in the lab, but shake flasks are used for small scale or for fundamental study. Okay, for example, if you see in Ahmad's story, he just isolated the microbes, right? And then he tried to grow the microbes. And the reason why he used shake flasks and not yet all those advanced platform is because shake flask is a standard practice for any basic fermentation in the lab. So if let's say we want to study something, we want to start investigating the growth of new microbes, we will adopt shake flasks first. So it's the benchmark. Okay, and from there, if let's say we need to grow in a larger scale, then only then we move to the bioreactors. Okay, so shake flasks are those platforms that are conventionally used in the lab for standard fermentation. So you know that shake flask or Ellen May flask. So in order to carry out fermentation, we have to put the shake flask on an incubator shaker. So that's what we call the machine, the shaker. So it provides the shaking to the shake flask and also the ambient temperature. Yeah? When we put it in a closed incubator, it means that we can set the temperature of the box to be at certain temperature so that the microbes can grow at that particular temperature. Okay, but the limitation of the shake flask, although it looks like very easy, yeah, you can just put the media, put the microbes in it, and then you put on the incubator shaker, and you can start the fermentation. But it doesn't have certain control systems, like there's no pH system, pH control system. There is no dissolved oxygen control system, and dissolved oxygen control system is important for aerobic because we can control the amount of oxygen to be inside the culture. So this thing, if you see this one, if let's say we put the microbes, you put the media into it, we call it culture, yeah? So that's the term that you have to know. So that shake flask doesn't have actually. And then in terms of the mixing, you know that during the fermentation, we have to provide mixing to the shake flask in order to make sure that the things are, the media are delivered to the microbes. There's no sediment at the bottom of the flask, so that's why we need shaking. Although we can provide shaking to the shake flask, but at certain point it might not be efficient, especially if let's say we have large flasks, large shake flasks. And let's say shake flasks, there are various sizes of it. They are 100 ml, 250, 500, 1 liter, and even up to 5 liter shake flask. But the bigger the size, when you put on the shaker, and the shaker, the incubator shaker, it has certain limit of speed. So we cannot provide like 1000 RPM. So I think the highest is about 300 RPM, like that. It depends on the brand as well. So it might not be that efficient to mix large volume of cultures. Okay, so that is one of the limitations of shake flask. So if let's say we want to carry out large scale fermentation, it might not be that really ideal to be carried out in shake flask. Okay, a limited scale of operation. 5 liter is, we don't carry out 5 liter, it's maybe up to just less than 1 liter actually for shake flask. And they are normally used for preliminary studies and not for production scale. So production scale is something, for example, at the factory, at the industry. Okay, we want to produce for the consumer's consumption, for example, that is production scale. So of course, at the factory, we don't rely on the shake flask, okay, because it's not practical. So because of that limitations, the scientists, the engineers, the bioprocess engineers, they have invented bioreactors, which have all of those control system, pH control system, temperature control system, dissolved oxygen control system, and then it has a better mechanism for mixing, right? And also there are various sizes from 1 liter bioreactor to even up to 100 liters of bioreactors, which are used in the factory. So because we want to overcome the problems of the shake flask, okay? So you can see that when we talk bioreactor is the general term. There are various sizes, there are various models, there are various brands. Yeah, same like any other products like car. There are many types of cars, right? Many models, many size. So same goes to the bioreactors. Okay, and how to know which bioreactor is suitable? It depends on what type of fermentation that we are carrying out, what kind of microorganism, what kind of products. Yeah, so those things come into play when we want to decide what type of bioreactor or fermenter. Okay, so what a bioreactor has and a shake flask doesn't have. So you see this one is a shake flask, and we put it in this incubator shaker. This is what we call incubator shaker. So some of you answered incubator shaker just now. Is not really correct, because what I mean just now is the fermentation platform is the one that where we contain the microbes, which is actually should be the shake flask in that question. Yeah, right. So shake flask, we cannot just put it on the table just like that. Maybe it's possible still for the fermentation to take place, but it will not be efficient, because at the end you can see that the media will sediment at the bottom. So it means there's no mixing. The microbes might not be able to get enough nutrients. So the fermentation might not happen effectively. Okay, some of you answer what is the limitation of using bioreactor. So I'll discuss it later. Okay, thank you for the question. For the shake flask, so I've mentioned there's no pH control during the fermentation. What you can do, you can just set the initial pH. So initial pH means that, let's say you want to set the initial pH of the media before the fermentation. But after you sterilize the media, you put the microbes, you cannot really check the pH unless you have certain way of doing it. But normally we don't check the pH during the fermentation. So we do not know at, let's say at 10 hours, what is the pH. We can withdraw some samples, check the pH using pH meter, but we cannot control inside what's inside the shake flask. Okay, so that's what I mean here. No pH control during the fermentation. The temperature control, we can set the temperature using this incubator shaker, but it just provides what is ambient temperature. We cannot control exactly inside the shake flask. Okay, so it depends on the environment temperature. So sometimes it might not be sufficient for certain, let's say for certain sensitive microbes. Let's say what inside the shake flask could be sometimes much higher, the temperature. So we cannot control the things inside. So in that situation, it might not be ideal for the microbes. And then there is no gas control. What I mean here is like we cannot control the amount of oxygen going in to the flask. Okay, so for the shake flask fermentation, for aerobic fermentation, it depends on the surface aeration. What is meant by surface aeration is the exchange of air that goes through this gauze. This is like gauze cloth, gauze, the one that we use for wounds, for wrapping wounds. So we wrap it with cotton, right? And then we make it like a stopper, and we put it on top of this flask. So from there, the air can go through the cotton and the gauze, and that's how the oxygen can come in. So you can see that it's very limited for aerobic fermentation. And for those microbes that are highly aerobic, means that they need lots of oxygen, shake flask might not be suitable. Okay, so that's one of the limitation. And for those that are not sensitive, might be is still okay for the shake flask. And then it's not connected to computer or mobile devices. Okay, yeah, you can expect it's just a simple apparatus like this. You can't do anything to connect it to the computer, unlike bioreactor, which I will talk about it later. And it's just used for small scale fermentation, suitable for small scale, not automated. You need to handle it manually. So means that you have to take one flask, you have to put it like, you have to do it manually, means that you have to add by yourself the media. But for the bioreactors, some bioreactors, they are automated, means that they have the system to put in the media, to take out the samples, like this kind of automation. But it depends on the brands as well. Okay, so all of these limitations are what the bioreactor offer. Okay, so what the scientists or the engineers they invented, they want to overcome the problems in the shake flask system. Yeah? They provide the pH control, means that there is a pH probe. Like this is the vessel, this is the what we call bioreactor, and it is connected to a control unit. Okay, and this control unit sometimes it is connected to I mean the modern one will be connected to the computer and to the mobile devices. So from there you can control easily what happened inside the vessel. What's the pH, what's the temperature from your mobile. So you can see the difference, right? And the pH control is because of there is a pH probe. pH probe is the one that measure the pH inside the culture. This is culture. This is culture. And then temperature control, there is a temperature probe. P-R-O-B-E. It's like it's not a sensor. It's like a device that can measure or read the temperature inside the culture. Okay, and the information will be sent to this control unit, and from this screen you can know what's the temperature inside the vessel, what's the pH, what's the oxygen level, because we can also control the oxygen to be delivered into the vessel, the culture. And then modern bioreactors are connected to mobile devices, to computers, right? And bioreactors are able to cater large volume fermentation. So this is not the only size. There are various sizes from even less than 1 liter. There is small bioreactor created nowadays. And there are up to 100 liters, more than 100 liters. If you go to the factories, there are various sizes of bioreactors depending on the application and the purposes. And then they are automated. What is meant by automated is that it's something related to connected to robots, to maybe like sensors. So it's to be done like you know, through computer automation. Okay. So I've asked a question during the forum on ELIP, right? What you would expect for fermentation in 20 years? So a lot of you answers quite good answers, like you have provided like technology point, right? And then the platform, the difference will be modern fermentation or future fermentation will have all these modern technologies like artificial intelligence. So those things are, yeah, that are possible with the modern bioreactors. And actually those things are already, I mean, they are already exist. Those technologies are already exist. The artificial intelligence have been used for fermentation, the platforms, the, you know, all these modern things. So, and of course, those things are evolving. So from year to year, the engineers, they will improve the system, the performance, like the connectivity, for example, the automation, up to a level that you don't have to really stay in the lab. Yeah, so that is possible because manually, like if we say about manual fermentation or something to do with traditional fermentation, what we have to do is like the person has to stay in the lab to take care of the microbes, to the culture, take the samples every 2 hours, for example. But with the modern bioreactors, those things will be done by the robots. So at certain hour, you just set the timing for the sampling. So every 2 hours, the robots will take the samples and it will analyze, give you the reading. So you can just monitor the growth of your microbes from your home. Yeah? So that's the difference between the modern bioreactors and modern fermentation and traditional fermentation. Okay, so that the features of bioreactor in general, and these features will be evolving. It's not just limited to this, and there are more to come in the future. Okay. Okay. So there is a question just now. You said that what is the limitation of using bioreactor? Limitation. Limitation. What are the limitations when it offers too many advantages. Limitation, I can say maybe if let's say, okay, let's say lah, if let's say we are too dependent on the technology, when something goes wrong, like maybe the electricity goes out, right? Power outage, that could be a problem. Limitation. I barely could see the limitation. Maybe yeah lah, something to do with those defects in the technology lah. Maybe if let's say there's something wrong with the electricity, there's something wrong with the connection, so everything could be affected. But you could see that modern bioreactors have more advantages compared to the traditional platforms. Okay. All right, so here we're going to look at different types of bioreactors. There are many types of bioreactors, and it depends on the type of microorganisms that we want to grow. It also consumes huge cost. Yes, correct. The cost is one of the bottleneck of the investment of the bioreactors. We need to invest lots of money for it. And yeah, I have to admit it's very expensive. And in Malaysia, the technology might not be that really high yet. So we have the limitations in terms of the types of bioreactors compared to advanced countries or modern countries like Europe, USA, Japan. So all the developed countries, they have high-tech, state-of-the-art bioreactors. Do we have bioreactors in university? Yes, we have bioreactors, but our bioreactors are quite old, I can say, not the latest one, but still it can carry out fermentation. So this is an example of bioreactor in Unimas, and this is, I have to say, is a quite old bioreactor. It's like maybe 20, 30 years ago, the technology, the model, I can say. Nowadays, there are more modern bioreactors. Okay, I will introduce the modern bioreactors in one of the LUs, I think in LU 7. That one concerns with the rapid technology in fermentation. Okay, thank you for the question. So bioreactors, the types of bioreactors, there are many types of bioreactors. So here we are going to look at the basic types of the basic designs of bioreactors, which are actually, even like the modern bioreactors, they have these types of bioreactors. This is like the basic, how to say that, the basic things about all the bioreactors, regardless whether they are modern or traditional. Just now the question how to clean the bioreactors, that one will be covered in the later subtopics. Okay, thank you for the question. So the first type of bioreactor is a stirred tank bioreactor or stirred tank reactor. So this is the schematic diagram of it. Okay, so why it is called stirred tank bioreactor or stirred tank reactor? Sometimes it's called stirred tank fermenter as well. STF, STB, STR is the same. Okay, so the reason why it is called stirred, because it has this agitator or this impeller. So this agitator or impeller, it provides the mixing. It will agitate the things, and it is connected to the motor. So when we connect this motor to the electricity, it will turn on and it will mix the things inside. So the whole system is actually not shaking. It's just static. It's just this thing that is moving. And that's how the culture inside it is mixed. Okay, so that's why it is called stirred tank. Stirred tank. The mechanism is by stirring. So is the most common type of bioreactor, and it is made from two types of materials, glass or stainless steel. So normally the one that is small, when I say small, is like maybe less than 10 liter. It is normally made from glass, but it's a special kind of glass. It's not the normal glass. It's, I think, borosilicate glass. So which means that the glass is very durable and resistant to high temperature, because this vessel needs to be autoclave, needs to be sterilized. So when we autoclave, we apply high pressure. So those types of glasses that cannot resist high pressure, it will break. Okay, so this one is it is made from a special kind of glass. Um, so so when the engineers design the bioreactors, they have certain configuration in terms of the height to diameter ratio. So what is meant by height to diameter ratio is this is height, the height, and the diameter is the diameter. So the ratio of the height to diameter, they are certain values that is used for bioreactors, which is between 2 to 6 to 1. Okay. So that is the rule of thumb. Right, so the stirrer or motor may be top or bottom. Okay, the stirrer is the one here. This is the whole stirrer or agitator are the same. Okay, they are connected to the motor. So in this case, they are top driven. Top driven means it is at the top. So some bioreactors, the motor is situated at the bottom. So in that case, it is bottom driven. Okay, it refers to the location of the motor. All tanks are fitted with baffles. So inside this bioreactor, there are many components actually, and one of them is baffles. Baffles is the one here. Baffles is actually just a metal that is inserted inside that in order to improve the mixing pattern. Okay, so this is something to do with the engineering part of the bioreactor. So why there is baffle? Because the engineers, they have discovered by adding certain type of metal inside at certain position, it can improve the mixing. Okay, and mixing is important for fermentation because we need the bacteria and the media to be mixed properly for efficient fermentation. Stirrers are fitted with agitators or impellers. Okay, agitators, impellers. This is impeller actually. So it will be actually rotating inside that. Okay, so that will drive the mixing process. And there are many types of impellers, many types of agitators. The most common one is the four-bladed is turbine. Okay, this is just for your information. Okay. And then another thing is A sparger must be below the stirrer. Okay, so there is when you have this one, you have to provide the air, right? Let's say it's an aerobic fermentation. So it is provided, the air is provided through a sparger. Sparger is the one is a tube that is connected to it. Now I will show it in the later slide. Okay, so there is a sparger, and what the sparger does, it has holes. So from there, we connect to the oxygen source, and from that holes, that is how the oxygen can be supplied to the culture inside the fermenter. Okay, so distinguish the mixing mechanism between shake flask and bioreactor. Okay, so you see this one shake flask we put on the incubator shaker, because if we don't put on the incubator shaker, there's no way for us to mix it. If we just put on the table, it will not mix properly, okay, unless you manually shaking it, but it's not possible for you to shake it for 24 hours. So you have to put it on the incubator shaker. So the mechanism, when I say the mechanism, is how the mixing takes place is by shaking. When you imagine something is shaking, is like something is in a circular motion like that, right? But for the bioreactor, the whole system is static. It's not shaking. I mean, the whole bioreactor is static, but what is shaking, what is moving inside is the agitator, which is connected to the motor, and this will be stirred inside. Okay, so let me show this animation how it looks like. So we don't need the music, maybe just to show how the mixing takes place in the bioreactor. Can you see this?
Yes.
Yes. Okay, that's how the stirrer looks like. Yeah? I mean, it's shaking inside. Okay, you can see that the whole system is not moving. The bioreactor, the vessel, is not moving, unlike the shake flask, which we shake on the incubator shaker. Okay, I just want to show that actually. Okay, let's go back to our lecture. Okay, that's the main difference between shake flask and bioreactor. Okay, tank bioreactor. Okay, so So this is the head plate, we call it. Yeah. So this one is, if you see here, this is motor. This is the one that drives the steering just now. And if you take this thing out, this metal, this is what we call head plate. Head plate. So it has many components inside. Actually, it covers like the whole thing here. So there are many inlets. Inlets means a hole whereby you can insert the probe. The probe is the device that, for example, the pH probe, the temperature probe that we insert inside the vessel. So on top of here, you can see there are many inlets actually. And then this is baffle. This is the metal that I call baffle. And then the whole thing here is this one is impeller. This one is the one rotating inside, yeah? Impeller, anti-foam sensor, temperature sensor, level sensor. Yeah, there are so many types of sensor. Okay, so what this level, temperature sensor, anti-foam sensor do? So temperature sensor, it will detect the temperature. It will read the temperature inside inside this one, inside the culture when the fermentation is running. For the level sensor, this one is to detect, you know, the level of culture, yeah, the level of the culture, so that if let's say it overflows, sometimes there is a situation where the culture overflow, so that level sensor, it will give the information to the control unit, and from there we know that there is something wrong with, there is something wrong with the level of the culture. So antifoam sensor is a sensor that detects the formation of foam. Okay, so and then from there it will direct the insertion of antifoam. Okay, I'll talk about antifoam later, but this is how the head plate looks like. Okay, right. So that is third tank bioreactor. So the second type of bioreactor is airlift bioreactor. So this one, the difference is in terms of the mechanism of the bioreactor. It doesn't have stirrer, but it depends on the aeration bubbles. Okay, so if you see that this is the bioreactor, how the bioreactor looks like, and there is a sparger ring at the bottom of this one. Okay, and this is where the air comes out. Air comes out from the source, right? And it will comes out in the vessel in the form of bubbles like this. And these bubbles will be directed upward, yeah, and throughout the reactor. And that bubbles actually drives the mixing process inside the vessel. So means that in this airlift bioreactor, there is no impeller, there is no stirrer, okay? So that's why it's called airlift. It depends on the air, the mechanism of air. So there are basic loop, internal loop, external loop. So there are types of airlift reactor. Okay. Right. And this one is very popular for growing plant and animal cells because plant and animal cells, they are very sensitive to shear force. Shear means like the force created by this whatever the mixing process. Let's say in the third tank reactor, the force inside is very strong actually because of the stirrer. So that reactors is not suitable for animal cells because animal cells are very sensitive to the rupture. Okay, so in that case we have to use airlift bioreactor. Okay, so that's why the types of the bioreactor, it depends on the types of organism that we grow. Okay, so this is examples of algae. Algae is a plant cells using airlift bioreactor. Okay, the disadvantages of airlift bioreactors, it requires high capital cost to build large-scale vessels, high energy cost to supply high air pressure because it depends on the air. So it means that the mixing, everything depends on the air. So we need to provide high pressure. So high pressure means high cost. Yeah. And then hard to maintain consistent levels of nutrient and oxygen in large vessels, formation of foam. Yeah, so those are amongst the advantages of airlift bioreactor. Okay, the third type is loop reactor. The loop reactor, it combines stirrer and also the airlift feature. So it means that it has the sparger, the formation of bubbles, but it is supported by the impeller at the bottom part here. Okay, So technically it's a modification of airlift reactor, but it has the turbine at the bottom. So it means that it's a little bit more efficient in terms of the mixing, but to use this kind of bioreactor, we have to look at the types of microbes that we grow. Some of the microbes are not suitable. Okay, but it's just for you to know that it combines the impeller part and also the airlift part. So this is loop reactor. I'll just go very briefly. You can read by yourself. Just highlight the important points. Okay, the fourth system is disposable system or single-use bioreactor. So this one will be covered in detail in LU7. So basically what this bioreactor is, they are made from single-use material. So that's why they call single-use bioreactor. So the single-use materials are plastics. Okay, you might be wondering why we want to create bioreactors using plastic. Okay, so there is a trend nowadays that in modern years of fermentation, the engineers, they create bioreactors using disposable materials like plastics because they want to avoid the cross-contamination. And especially when the production deals with expensive products like vaccine, like antibodies, or something that they do not want to take a risk of contamination. Why contamination? Let's say you use glass bioreactor, you can use it many times, right? But the problems with the glass bioreactor or the stainless steel bioreactor, from one batch to another, you have to clean it. So between the batches, if let's say you don't clean the bioreactors carefully, there might be trace from the previous batch. So it might be contaminating the next batch. Okay, so that's one of the disadvantages of using glass or stainless steel bioreactors. So in pharmaceutical industry, they do not want to incur that kind of risk. So what they do is that they invest on using single-use bioreactors, whereby the bioreactors are made from plastics, brand new plastics. So every single fermentation of every single production, they would just use new bioreactors or these single-use bioreactors. So from there, they are very sure that they would not face any contamination problem. Yeah, and that is very worth because they are dealing with expensive products like vaccine, like pharmaceutical products. So single-use bioreactors, they are mainly used for those industry, pharmaceutical industry. Okay, so that's why the engineers or the scientists, they created this type of single-use bioreactor. So we can talk about it in detail in LU7. Okay, but at this point, it's enough for you to know that there are these types of bioreactors which are made from single-use materials. And then the last type of bioreactor is immobilized system. This one is, as the name implies, some part of the bioreactor is immobilized, means that they are not moving. Okay, so this is the one. The beds is made from porous beds where all the cells are trapped into it. Okay, so just now, when we talk about stirred tank bioreactor, the whole thing are moving freely inside the vessel, the culture, right? But this one, there is an immobilized part over here where the cells are trapped, and what is moving, only the media is moving throughout these bags. Bags is like the small parts that trap the cells. Okay, so microbial cells are attached to a solid surface made of gel, glass, or plastic beads, and then this organism need a surface to attach for growth. Yeah. So the reason why this system is introduced, it is used for those microorganisms that are sensitive. Means that if let's say they are grown using stirred tank reactor, the cells might be broken easily. So that's why they need to be immobilized. Okay, so this system is designed for that particular type of microorganisms. And also sometimes we use this system for enzymatic reaction. So bioreactors or fermenters, they can also use for enzymatic reaction. Enzymatic reactions are not fermentation because we are using enzymes. We're not using living cells. Okay, so this system is also suitable for enzymatic reaction. Okay, and also for plant and animal mammalian cells. Okay, right. So this is immobilized system. So you just remember that this part is the immobilized part. Okay, so we have covered the types of bioreactors. The next part is the fundamentals of bioreactors. Now we are going to focus more on the stirred tank fermenter, because the stirred tank fermenter is the one that is widely used in fermentation. You guys need a break?
Yes, doctor.
Okay, 5 or 10 minutes. 5 minutes or 10 minutes?
10
5
minutes.
minutes.
10 minutes, doctor.
10 minutes?
Yes.
Okay, 10 minutes. Okay.
Thank you, doctor.
Thank you, doctor.
Here. I'll see you back at 11:55.
Okay, so
Okay, let's resume.
yeah.
Okay, the second part is on the fundamentals of bioreactors. I will just go very briefly on every point. I'm not going to go into detail. Okay, so I just guide you some of the important points. So here in this second part, we are focusing on stirred tank bioreactor a lot. Okay, because that's the most common bioreactor for microorganism. Okay, so first is when we talk about bioreactors, what's the important parts? What are the important aspects of bioreactors? We need to know the design of the fermenter vessel. So what kind of things that we need to consider when we want to talk about the design, the geometry, okay? The geometry. So normally the bioreactors, they are in vertical cylinder. What is meant by vertical cylinder? It is tall in nature. So the reason why, because it will save the space rather than to have vertical fermenter. Vertical fermenter, not vertical, horizontal fermenter. So horizontal fermenter, it will take lots of space. So normally the design is vertical. So it doesn't occupy more space for that. Okay. And also if you see the bioreactor, normally at the bottom, you would see that it has concave shape. Concave shape means that it is not, there is no sharp edge. Okay, so the reason is because that will ease the cleaning process. So there will be no residue stuck at the edge of the bioreactor. So that's why the vessels are normally concave in shape. Okay, so that's the design. And then the body of the vessel, you can see that when we fill in the bioreactor, the vessel, this is what we call vessel. The whole system we call bioreactor. Vessel is only those that is the container or the glass part or the stainless steel part. So when we fill in the vessel of the bioreactor with a certain amount of media and then we put in the microbes, yeah? So the whole thing here, it is called working volume. The one that is only covers the media, the microbes, whatever in the suspension. So this is working volume. But when we talk about reactor volume is the whole volume of the bioreactor here, the vessel. So we cannot fill in to the maximum because it will create pressure. I mean, we need some space actually. So this space is what we call head space. Yeah, and normally the allocation for the head space is about 20%. It cannot be less than that. It cannot be just 10%. Otherwise, it would not be enough space for the pressure. Okay, so the rule of thumb for the working volume is to be 80% of the total reactor volume. Okay, so now you know what is working volume. So this headspace function to prevent contamination, provide a gap, allow the formed air bubbles arising from within the broth, okay, allow back pressure of air to build up. Okay, so during the fermentation, there are air coming out or bubbles produced. So that headspace will allow that formation of air bubbles and also form. So that's the importance of the headspace. Okay, so yeah, take note the terms reactor volume, working volume, what is headspace, what is the rule of thumb for the headspace, which is 20% headspace, 80% working volume. Okay, and then when we talk about the design of the bioreactor, we are also concerned with the materials for the vessel construction. Okay, this is vessel. This is what I mean by vessel. There are many sizes of vessels, many sizes of fermenter, and also the materials, it could be either glass or stainless steel. I mentioned about it just now. So the glass, the type of it is borosilicate glass. Borosilicate is very resistant to heat and also pressure, high heat and pressure, because we autoclave the vessel. So when we autoclave, we apply high temperature and also high pressure. Okay. Normally, the glass vessels are limited to small scale fermenters, which is maybe like less than 10 liter. Okay, so for more than 10 liter, normally we use stainless steel as the material for the bioreactor. So the reason why stainless steel, because it can cater better performance for large bioreactor. It's strong, yeah, robust, and then it's ideal for large construction, yes. And then, yeah, but the whole thing here, actually, when it is made from stainless steel, it's not actually totally stainless steel that you cannot see inside. There is a side that you can see what's happening. So here, I don't have the picture, but this is what is meant by here. Glass is used for observation pots. So there is a point where there is a glass and you can see the level of the culture inside the stainless steel bioreactors. Okay, so those are the two materials used for vessel construction, glass and stainless steel. And then another part of the bioreactors is agitation. It's very important for stirred tank reactor. So this is how it looks like from top view. This is what we call impeller. This is the impeller. The drive here is shaft. This is the long, is the one that connects the impeller to the motor. Okay. So the agitation, the types of the impeller also plays an important role, right? There are many types of impeller. This one is, I think, propeller or tank. Yeah, there are many types. Here are all the types. What's here? The impeller. Okay, so these are the types of impellers. Okay, many designs of impellers. How to know which one is good is based on the research. But the most common one used is the Rushton turbine. This is just for your information, not really. It's more to engineering part lah, actually. Yeah. So enough for you to know that there are many types of impellers. Okay, which can cater many applications or many purposes. Okay, go back to the point here. Yeah, that's impeller, which is inside this one. Okay, so the drive configuration. So the drive configuration, this refers to the location of the motor that rotates the shaft and also the impeller. Okay, so there are two types. One is bottom driven. Okay, you can see that the motor is located at the bottom of the vessel. This is vessel. And then the second type is top driven. Top driven is the motor is located at the top. Okay, both of them have advantages and disadvantages, and it depends on the application, depends on the purposes of the fermentation process. Okay, some of the fermentation process it requires bottom drive reactors, and some requires top drive reactors. Okay, so the advantages for the top drive here. It is not really exposed to leaking, because this one motor, it can cause some sort of misalignment sometimes, and when that happens, especially for the bottom drive, it could cause leaking at the bottom. So that's the disadvantage of bottom drive compared to top drive. For the top drive, it doesn't have that sort of risk. Okay, so that's one of the key advantages of top drive. The rest you can read by yourself. I can just skip some of the points because I think I don't have enough time to cover all in detail. Okay, so agitator types. All right. Okay, no, just now we talked about the different types of impellers. Impellers is the one here, So apart from the types of impellers, the distance from one impeller to another impeller. Okay, this one impeller here is one impeller. So if let's say you have large reactors, you may need more than one impeller. Sometimes it could be 4 to 5 impellers, depending on the height of the vessel. So the distance here, it plays an important role as well. It's not just the type, because when the shaft is mixing, when the shaft is rotating, okay, so the impeller, it will create certain pattern of the mixing, certain pattern of the fluid inside that. So if let's say the impeller is not distanced properly, yeah, maybe it's too far or too close, so there could be what we call dead zone inside the reactor during the fermentation. So when there is a dead zone means that that part is not mixing, means that the media in that part is not circulated. So means that, or maybe the cells at that part is not circulated well. So it could lead to inefficiency of the fermentation. And so that's why for when we talk about the impeller types or the agitator, the types of the impeller plays an important role, and also the distance of the impellers on the shaft. This is shaft. This long stick is the shaft. The distance means the distance from one impeller to another. Both play important roles. Okay, and apart from the speed, okay, the speed, of course, you can control the speed of the shaft of the motor during the fermentation. And how to know what is the speed? It depends on the types of the organism that we are dealing with. For bacteria, normally they are not sensitive to high speed, so we can apply high speed. But for certain microorganisms that are very sensitive to shear force, we need to just apply a mild speed. Mild speed means like it depends on what has been reported for that type of microorganism. Sometimes it's less than 500 RPM. RPM is the unit of the speed. Okay, so the speed also plays an important role during the fermentation, the speed of the agitation. Okay. So what else? This is the types of impellers that I've mentioned just now. Right, apart from agitation, aeration. Aeration is also an important aspect. What is aeration? Aeration comes from the word air, actually. It's the provision of air inside the bioreactor. So this one is very important for aerobic fermentation, because aerobic microorganism depends on the oxygen to grow, right? So if let's say we don't provide enough oxygen, the microbes might not grow well, and it might be dead. So how are we going to supply the air, the oxygen, to the culture? We have to sterilize the air. So how we sterilize the air? By using filter, air filter. Okay, so this is how the bioreactor looks like. This is sparger, we call it sparger. The whole thing here is connected to this loop, and it has small holes, where this is actually connected to the source of the oxygen. So when the oxygen comes into this, so it will go out through these small holes inside this culture, and you can expect that bubbles will come out from that holes. Okay, so that's how the oxygen is supplied to the culture during the fermentation. Okay, so that's how it looks like. And then, so for aerobic fermentation, we supply oxygen, but for certain cases, we might also supply other types of gases. For example, anaerobic fermentation. Anaerobic means it doesn't require oxygen, right? So in order to eliminate the oxygen from the culture, we sparge nitrogen, because nitrogen can eliminate the oxygen. So for the anaerobic cultures, we sparge nitrogen using sparger, and what comes out is the bubbles of nitrogen. Okay, so that's why in some cases other kinds of gases are sparged into the bioreactors. But for the aerobic, it's oxygen or air, because air consists of oxygen. Okay, so aeration is a very important aspect in bioreactors. Okay, so you know about aeration already. So talking about the transfer of oxygen, okay, so this is the gas bubbles that I mentioned just now. When the gas bubbles exist in the suspension in the culture, actually for the oxygen to reach the microorganisms, it has to undergo a few layers or a few phases. Okay, so this is what is described by this diagram, whereby the first phase is the gas phase. So this is gas. You imagine the bubble, it is actually gas. And this is liquid. The whole thing here is liquid. So it has to undergo the layer between the gas and the liquid, the gas-liquid interface, like this one, number 2. And then number 3 is the liquid film around the bubble. So there is a layer called film. liquid film. And then that's what the oxygen has to undergo. And then it has to undergo number 4 is the liquid, the culture medium. And then 5 is the liquid film around the microbial cells. So this is the cells. The cells is like the solid, the intermediate is to be solid. And then this is another film. And then 6 is the cell liquid interface. And 7 finally is the inside the cells. So that are the routes of the oxygen from the gas bubbles to the microorganism. Okay. Right. The details of it you can read by yourself. So this one is solubility of oxygen. Okay, when we talk about solubility of oxygen, we supply the oxygen to the suspension, to the cell suspension, which is in the form of liquid. Actually, it's not that straightforward. Straightforward in a way that there is a limitation. Not all of the oxygen will be actually taken by the cells. So those that are taken by the cells that are only the dissolved oxygen, oxygen that can dissolve in the liquid. Okay, so when we measure the level of the oxygen, we measure by using this device which we insert in this vessel. Okay, this one is to be inserted here. And then what this device will measure, it will only measure what we call dissolved oxygen, not the total oxygen, because it cannot measure the one that is not dissolved. Dissolved means that the one that is dissolved in the liquid. Yeah, because we talk about two different phases. Oxygen is gas, and this culture is liquid. Okay, the one that is that can dissolve is the one that is measured by this probe. We call it DO probe. Okay. Yeah. So by nature, actually, oxygen is not that really easy to be dissolved in water. It can, but it takes lots of routes actually to be dissolved. Factors affecting oxygen demand. Okay, is the rate of the cell respiration. So it depends on the types of the organism, okay, whether it is highly aerobic or not aerobic or facultative aerobic, moderate aerobic. And then the types of respiration, aerobic, anaerobic, yeah, type of substrate, glucose or methane. Okay, the type of the media supplied to the organism, it will also influence the way how the cells respirate or how it metabolizes, the behavior of the cells inside the culture. Okay, the type of environment, and all of this will affect the oxygen demand. Okay. What else? To ensure the balance between oxygen demand and supply, we must make sure that the oxygen uptake rate of the culture does not exceed the oxygen transfer rate. Okay, there are two things here. The first one is oxygen transfer rate, is the amount of oxygen that we supply to the culture. But doesn't mean that all of them will be taken, right? So oxygen uptake rate is the amount, the rate of the oxygen taken by the microorganism inside the fermenter. So it means that logically the transfer rate should be more than the uptake rate. Okay, the one that we supply should be more. Otherwise, if let's say we supply less than what the microbes need, then the microbes will die. So the OTR, this one is normally denoted as OTR, this one is OUR. OTR should always be more than the uptake rate. Okay, by logic, you can also think about it. Okay, there are so many works, actually, research that have been done to study about the oxygen transfer during the fermentation by different organisms. So these are reported in various research papers. Okay. Right. Another fundamental aspect of bioreactors is the heat exchange. So how the temperature is controlled. So if you see the vessel, so this is the vessel, you can see that there is actually a layer whereby it is not a single layer. It's actually there is a double layer. So this double layer is what we call a water jacket. So this water jacket, it means that is how the vessel, the temperature of the vessel can be controlled, where the water is channeled inside this water jacket. This is what we call water jacket. inside this water jacket, and there is an outlet that takes out the water. So the cooling and heating takes place in this water jacket. So that is how the temperature of the vessel can be controlled. Okay, so this is something to do with the mechanism of the temperature control of glass vessel. That's one of the ways. Another way is by using this cooling coil. You know, like when you have the kettle, there is a coil inside, and that coil is actually the one that plays a role in heating, right? So some of the bioreactors, they have this coil instead of the water jacket. Okay, so I'm talking about the mechanism of the heat exchange or the temperature control. Okay, and then pH control, that's one of the important aspects that differentiates bioreactor and shake flask. The bioreactor has this pH, this is actually pH probe. So it can be taken out and in from this top plate. If you see these red items here, red stuff here, this is actually the head of this pH probe. So it can be taken out. And then that is the one that measures the pH of the culture inside. So it will be hanging actually inside this bioreactor, whereby it will touch the suspension, and from there it can read the pH of the culture. And this one is actually connected to the control unit, and you can read the pH on the screen of the control unit. Okay, and the pH control, how the pH of the culture inside the bioreactor can be controlled. Let's say we set the pH to be set at, for example, 7. But sometimes because of there are so many products, organic products produced inside the culture tend to be acidic inside. Okay, so what happened? So when the pH probe detects the pH inside is acidic, it will give the information to the control unit, and it will give the instruction to control the temperature. So how the control takes place is based on the addition of acid or alkali. Okay, so if let's say the pH here inside here is acidic, you want it to be 7. Acidic here, let's say it is 3 or 4, but you want it to be 7. So you have by logic, it has to be added with some alkali, right, in order to go back from 3 to 4 to 7. Okay, so the alkali, the base, will be aided, will be aided based on the instruction given by the control unit lah. So it's something to do with the control. So once it detects the pH below 7, it will add some alkali until it reaches 7. So when it reaches 7, it stops the addition of base, and vice versa. If let's say the pH is basic, is more than 7, and then if let's say you want to make it like back to 6, so you have to add acid a little bit, but it will be automated. So that's the importance of automation for bioreactors, because it will happen by the automation. It will be controlled by the bioreactor. You don't have to add the acid by yourself, you know. So modern bioreactors, it has all these kind of things. Okay, so if pH is too low, yeah, so the pH control, basically the pH control is based on the addition of acid or base to the bioreactor. Okay, another things that you need to also consider when we talk about bioreactors, the sterility, how good is the seal. It's a closed system, so it means that the bioreactor needs to be sealed correctly in order to avoid the incorporation of organisms from outside. Okay, and then user friendliness. Of course, modern bioreactors nowadays will have all the user-friendly features, and it has lots of connectivity features compared to the traditional bioreactors. Flexibility, more flexible for the modern bioreactors. Okay, so those are other things. Okay, the second last part, the sterilization. How we sterilize the fermenter So in the previous chapter, for the LU2, we talked about media, right? So the media also need to sterilize. So that's why I asked what is the importance of autoclaving of the media before the fermentation. Is because we want to make sure that the media is clean, right? Without any contaminants. So the same thing goes to the fermenter. So once we prepare the media, we insert the media into the bioreactor, and the whole media and the bioreactor needs to autoclave before we start the fermentation. So how are we going to autoclave the bioreactor? There are a few ways. This is just sterilization in general. Okay, but for the bioreactors, we autoclave the bioreactor. So there are two ways of autoclaving. For the small scale bioreactors, we can use this type of small autoclave that looks like a washing machine, right? I'm sure that you have seen these autoclaves in the lab. So up to, I think, 2 liter bioreactor, you can insert the vessel inside this autoclave, okay? But if let's say the bioreactors are too big, more than, sometimes there are autoclaves that can cater, bigger autoclave that can cater bigger fermenter. But more than 10 liter fermenter, you have to use in situ sterilization. You don't use autoclave anymore. Okay? For example, like this kind of big bioreactors, it requires in situ. In situ means that this bioreactor is connected to steam system, whereby the steam will go through the bioreactor and it will be sterilized at that place. That's why it's called in situ. Yeah. So it's, we don't need we don't require autoclave anymore for large scale bioreactors. Okay, so there are two types of sterilization. One is autoclave for small scale bioreactors, and in situ steam sterilization for large bioreactors. Just now I've talked about aeration. So it's another aeration. Okay, for the aeration, so just now I mentioned about the provision of air inside the bioreactor, right? So that air needs to be sterilized. And how to make sure the air is sterilized? We cannot autoclave air. So what we do is that we have the tubing attached to filter like this. Okay, this one is an example of squat bottle, but then this kind of filter we attach to the tubing that connects to the bioreactor. So that, for example, that we have the source of air, and then that air has to go through the filter, and after it has filtered, then it will go through this vessel. Okay, that is how the air is sterilized for fermentation. Okay. So, and for the filters, there are many sizes of filters. It depends on the pore size, right? So if let's say we want to filter the air so that the air doesn't have any microbes, so we have to make sure that the pore size is less than the size of maybe the bacteria, the smallest bacteria. So the pore size plays an important role in the selection of the filter. Okay, so so the key point here is that for the air, it is sterilized by filtering the air that comes in to the bioreactor. Okay, I try to simplify the points that you have to take note for each of this part. Okay, I think this one you can read by yourself. For the sterilization, there are two types of sterilization: batch sterilization and continuous sterilization. It's kind of a little bit similar to the concept of batch fermentation and continuous fermentation. Batch sterilization means that you just put everything in, sterilize, and take it out once it is sterilized. But for the continuous sterilization, the sterilization is continuous. So there's coming in, there's coming out. Okay. I expect you to read by yourself this thing. And then the last part for the forming. Okay, during the fermentation, when the things is agitated, the cells are growing, proteins are produced. You have to expect that there would be form is buoy, that will be formed on top of the culture, okay, like this one. like this one. Okay, but this one is inside the bottle. But it happened. It happened in the fermenter. So the formation of foam, that foam is actually by nature, it is not desirable. Why? Because whenever there are too many foams, it will lead to the flow out of the culture out to outside. So it means that it would be overflow of the culture. It will be, there will be contamination. Okay, so that is what we don't like about foam during fermentation. But it can't be avoided because the nature of the fermentation broth, it produces protein. So these proteins is the one that contributes to the formation of foam. Okay, although it can't be avoided, but we can control how the foam to be in our culture. Okay, so there are a few ways how we can reduce the foaming. The first one is by having a headspace. So means that if let's say we allow certain enough space inside the bioreactor, we talk about headspace just now. So the maximum headspace is 20%. Sorry, the minimum headspace is 20%. It couldn't be less than that. But if let's say you want to increase to, let's say, 30% headspace, means that more room for more space on top of the culture. So it would be better because it means that we can have more space for the foam if let's say the foam are there. Okay, so this is one of the basic ways how we can reduce the foam by having enough space on top of the culture. Okay. But sometimes it might not be favorable because if let's say we want to reduce the culture, means that if let's say we increase the headspace, we reduce the culture, reduce the amount of the working volume. But sometimes we want to produce high amount of things. So we have to compromise between two, right? So if let's say we want to carry out large fermentation, large volume of fermentation, sacrificing the culture growth might not be suitable. So we have to find other ways how we can control the form by not, maximizing the headspace. So other ways how we can reduce the form is by using antiform. Antiform is the chemicals added that can control the formation of form. Okay, and normally those chemicals are oils, animal and vegetable oils, silicones, glycols. So there are specific chemicals that are normally used for controlling the form during the fermentation. So normally the tubing, and there is a tubing that connects to the antiform actually. So just now when I introduced the top plate, there is a level sensor, antiform sensor. It's form sensor. So that sensor, it will detect the level of form. So whenever the form is too excessive, so it will give the instruction to the control unit to put in the antiform. Okay, so that's how it works during the fermentation. The disadvantages of antiform, it could decrease, it could affect the oxygen transfer. So that's the disadvantage of it. And also it could affect the separation and purification of the product, because now we have additional stuff inside the culture broth, which is the oil, all these chemicals, right? So that is the disadvantages of it. Okay. Another way how we can control the forming is by using mechanical antiform. So what is mechanical antiform? We have this additional stuff, additional device where this one can break down the form on top of the culture. Okay, so this is what we call mechanical antiform. But the problem with this, okay, you can see that every approach, it has its own advantages, it has its own disadvantages. So it depends on our type of organism, it depends on our purpose of fermentation, how we want to compromise those advantages and disadvantages. Okay, for the mechanical antiform, the disadvantage is that it doesn't work at low RPM. So it means that because it needs high speed for it to, to operate efficiently, because it is something to be installed on the shaft over here. So it doesn't work at low RPM. So if let's say you want to use it, but you need to apply high speed. Okay, so if let's say you want to apply it for high shear sensitive organism, it might not be suitable because high speed might not be suitable for shear sensitive organism. So back again, it depends on the types of organism. It depends on the purpose of the fermentation. Whenever you want to adopt which way you want to use, either you want to use chemical endoform, mechanical, or maximizing the headspace just now. Okay, so that's the three ways how we can control the form during the fermentation. I think that's the last topic. That's the last subtopic. Any questions? Sorry for going too fast. So basically in this LU, we cover what is bioreactor, yeah, why we need bioreactor, the difference between bioreactor and traditional platforms for fermentation, that is shake flask. But not to say that we would just use, in modern years, we would just use bioreactors. No, we still have to do, we still have to use shake flask as the benchmark, as the starting platform. But you can see that as the application or the needs changes, yeah. So that is when we try to transfer the production, the fermentation process from shake flask to bioreactor. Okay, so we have covered what is, some introduction on bioreactor and the shake flask, the differences between both platforms, and then we have covered the types of bioreactor, 5 types of bioreactor. Fundamental of bioreactors, what are the important aspects when we talk about bioreactors? Sterilization, yeah, sterilization of the fermenter, how we sterilize it, how we sterilize the air that is connected to the fermenter, and also the media. And the final part is forming, how we control the forming inside the bioreactor. Okay, so the 3 ways of forming control. So that's all for today's topic. Any question?
So far, no, Doctor.
Okay. Okay, I hope you understand. You got more better ideas about fermentation, media, and now we talk about platform. Okay, so please remember from now onwards, when I say platform, fermentation platform is a place where you contain, where you put the microbes and the media for the fermentation to take place. So that's why incubator shaker is not the platform. It's not considered as the platform. Okay. So try to familiarize with all the terms, culture, growth. Growth is what? Bioreactor, fermenter. Okay, all right. So I think that's it for today. Thank you very much for your attention. Anyone needs manual attendance? I think some of you have scanned. Right, just will drop your metric numbers here. Okay, I'll see you guys in the next session.
All right, sure. Thank you, Doctor.
Okay.
Thank you, Doctor.
Okay, thank you, Doctor.
There's no mid-semester exam. There's one of you who asked the date of mid-sem. There's no mid-sem exam. You can refer to the Elite Man page. What are the assessments? Okay.
Thank you, Doctor.
All right. Okay. Have a nice day. Bye.
Thank you, Doctor.